Well, thank you for staying till the end of the conference. I promise it will be short. So now I'm going to talk to you about one of the most challenging problems in clinical trials, if not the most challenging one, which is drug efficacy. And so average failure rates in clinical trials from 2009 to 2018 reached 84.6%, which is like the highest number it reached, actually, like the failure rate. I mean, the highest rate of success that we've reached so far. And if you look at the right side, if you look at those pie charts for the reasons behind the failures in clinical trials, you see by far efficacy is the highest, is the biggest reason behind those failures. And these are the purple parts of the pie charts. And that is still the same reason until today behind those failures is those drug efficacies that are not high enough, or they can't pass the statistics that are required for the FDA approvals. So, and that is not just in certain disease area, but this is across different therapeutic areas. And despite the fact that we've made a lot of progress in terms of lead optimization in finding new molecules, and like we've seen in the past presentation, how far we've done and we've gone in that direction, we still see low efficacy rates in clinical trials, and that's to show that the real roadblocks is more than just the chemistry, and we need to also think about the target, the efficacy of the target, and this is very challenging. So in order to for me, before we get into the approach we're taking, we want to also get to understand why targets fail in efficacy. And one of the reasons, or two main reasons, correlation. So a typical scenario, you would find a protein that's highly expressed among patients, and it's not very highly expressed among healthy controls. So we would think, oh, this is a good target to go after. We design a drug for it, we take it to clinical trials, but still fails. And that's because this target is correlated with disease progression, but it's not actually causing the disease progression. So even after the drug binds to the target, the disease continues to progress. That's one scenario. The other scenario is that your target is causal, but it is far downstream from the origin of the disease. So by that time, the disease progression have branched out into parallel processes or parallel pathways. So yes, you are interrupting the cascade of the disease, but there are other parallel pathways, as you can see here. As you can see here, like there are different ways the disease will continue to progress. And if you hit only one of those pathways, you won't end up with high efficacy. So can we use AI to solve that problem? And if you look at those statistics, which were published very recently, actually, and it is quite exciting for phase 1 clinical trials. So you see out of those, 24 completed clinical trials in phase 1, 21 succeeded. And these are for AI-assisted drug programs, whether it was AI repurposed molecules or AI discovered targets or AI discovered molecules, which is quite exciting, actually. Like 21 out of 24 succeeded. It's pretty good for where we are today. But when you look at phase 2, the number decreases, and we still see more failures than, as you can see here, you still see more failure than successes. So 4 to 6. So, and the reason behind this is most of the AI approaches today are pure data-driven approaches. And it can work in a lot of cases, but in biology operates under highly regulated and precise mechanisms. So pure data-driven approaches do not benefit from the wealth of knowledge that we accumulated on systems biology. So we decided to go for a hybrid approach. And And the hybrid approach is where you use the power of machine learning, you use the power of data and big data, but also you fuse that with systems biology and the mechanistic models. So we build our disease models based on we use machine learning to build a comprehensive network of interactions for each disease, where we use the 3D structure of the proteins and other molecules that interact with proteins within the tissue to predict those interactions within the proteins themselves, or proteins and lipids, or proteins and metabolites, and so on. And once you've modeled those interactions and you have a full view of the interactions that happen within the tissue, we simulate the dysregulation that happened within the disease. And then we go into a simulation mode where on a simulation mode, we can simulate that intervention that the drug actually causes in those pathways. And then what we do is we use perturbation networks and to understand what happens downstream from that. So basically, you're looking at the effect of your drug on all the pathways all together, and also you're able to quantify that all the way downstream down to the clinical results for the disease endpoints. So the idea here is to be able to validate your target, give it a quantifiable measurement of efficacy, compare it to other targets that are already approved, for example, like drugs that are already approved, and know where you stand in terms of chances of success or failure in the future. You can also use a very similar method that we designed to also find signatures that would define which subpopulation of patients would be more responsive than others. And the idea here is that we're not using drug data, and that's why it's not very data hungry, and a lot of presentations today touched on a data problem. We designed this approach so that it's not data hungry. We don't need to test on on data from other drugs, but we just need to understand the disease more. The idea here is to understand the disease. Once we have a full view of the disease, you can predict the efficacy of novel drugs that haven't been tested before. So here, this is a screenshot of our platform for the target validation, where you see like the different interactions in different colors. So the blue is physical interactions that were tested in the lab before and were experimentally proven. The yellow is the interactions that we predict based on the binding sites of those proteins and based on the 3D structure of those proteins. And then you see also the yellow dots and the yellow lines, which shows how connected your target is to the disease endpoints, or at least what we define as molecular endpoints. For example, cancer, those molecular endpoints would be cell proliferation. And the idea is to get that quantified or quantifiable measurement of your efficacy, which is the score that you have here. And that, again, as I said, like you compare it with other drugs that are approved, and then you have an idea beforehand whether this is going to work or not. So we tested that actually against past clinical trials to see can we predict the results of those clinical trials based on those targets. And we tested that in 5 different diseases. And we predicted the results of those past clinical trials with 70% accuracy, which is pretty good compared to the 90% failure rate that we have today. And then the other algorithm that we've developed as well is the omic signatures. So it's also based on disease dynamics and disease pathways. But here we're focusing on different populations. So with the traditional biomarkers that we know is usually a single molecule or a single protein that is highly expressed in different subtypes of patients. But in this approach, we went a step further to look at signatures as actually a combination of changes that happen together. Because we realized that sometimes it's not just one change that is causing, it's simultaneously having multiple changes that actually drive a certain outcome versus another. And using this approach, we were more predictive of clinical results than using the traditional differential expressions approaches. So this we use to find biomarkers to separate or to differentiate between healthy and disease, or to differentiate between different states of disease progression, or to differentiate between responders versus non-responders. So using those two tools, we were able to cover most of the clinical, most of the stages of the clinical development of a drug, starting from discovery, from target discovery, validation, competitive landscaping, and plus the mechanism of action, because as you've seen in the disease pathways, there is actually it's a very transparent process where you understand the mechanisms that led to this high or low efficacy that the score is giving you. And we're also doing biomarker discovery and patient stratification based on the signature, the omic signature algorithm that we've developed. So this is a case study that we did on patient responsiveness in breast cancer. And the question we are addressing here is, can we predict drug responsiveness using samples or tumor samples from pre-treated patients? So the idea here is to predict if patients will respond or not from their tumor samples before they take the drug, before they've been treated. And to give you background, this is based on a study that was published in 2020, I-SPY2 clinical trials. In those multiple series of clinical trials, basically they tested multiple breast cancer drugs, and they collected their transcriptomics data from the patients from the pre-treated samples. And what happened is, so in this specific paper, they looked at inhibitor, like AKT inhibitor, drug designed by Merck. And basically, this drug, 35 patients responded to that drug out of 96. And the question they asked is which subpopulation of patients responded versus the ones that didn't respond. But it was very hard to answer this question, because if we look at the different subtypes of breast cancer, triple negative, HER2 plus, different subtypes, you see almost the same variation between responders and non-responders. So there was no clear answer why those specific 35 patients responded to that drug. It had nothing to do with a mutation behind the disease in those patients. So the conclusion on this paper was that they couldn't find a predictor for the response of the drug, but the response was good enough that the drug actually graduated and went to the next phase. So we took this data, independent of that study, and we decided to see if we can find predictors from this same data set to see, can we predict the drug responsiveness. So we analyzed the transcriptomics data from those patients, and we integrated it with the disease models we have for breast cancer with disease pathways to see, can we find signatures? And we found signatures. We actually found 2 signatures specifically that were significantly associated with lack of response. And we used this on a test set of the patients to see, can we predict the responses of those patients that weren't part of the training set? And we were able to do that. And our RC score was 0.71. So it was much better than using differential expression, which is the yellow line. If you just use differential expression, you're almost at a chance level. You can't predict the result. So the conclusion here was, yes, we can predict the patient responsiveness, and yes, that's only be from tumor samples, from their biopsies before they were treated. And then we decided to look at those signatures and see, okay, are these like random signatures, like random expressions that have nothing to do with disease? So we mapped them according to the disease pathways, and the red dots you see here are basically the famous pathways for breast cancer, MAPK1 and PI3K, and the different pathways that are known in the endpoints related to them. and the signatures are the ones in yellow. So you could see that they are very well connected to them. So it made sense contextually from a disease perspective, from also a mechanism of action perspective, that those signatures are associated with low response to that drug. And I just want to acknowledge that this work has been done by our team that you can see here. And yeah, that's the end of presentation. Thank you for listening.